The background leading up to this study is that there was some evidence suggesting that heat or increased temperature potentially interferes with spermatogenesis, or the process of developing new sperm in the testicles. I should emphasize, I'm definitely not a doctor, definitely not giving you medical advice, definitely not telling you what to do or what not to do if you're looking to have a baby or trying to avoid having a baby. These are just kind of my thoughts on the data and the study. When it comes to individuals trying to have a baby, sauna use may be an important factor to consider, in that if the male in the relationship is using the sauna, it may be impacting their sperm metrics and potentially their fertility or the probability that they'd be able to get their partner pregnant. Hey everybody, welcome to another episode of the Talking Study series, where I break down interesting research papers that I've read. I figure at the beginning of each of these episodes, I should probably start mentioning that it's important to take an overarching body of evidence into account when looking at any given topic, and that you shouldn't over extrapolate or overemphasize any one individual study. With that said, the study we're going to look at today is one called Seminal and Molecular Evidence That Sauna Exposure Affects Human Spermatogenesis by Garola and colleagues. And it takes a look at whether sauna use impacts a bunch of different sperm metrics and potentially male fertility. Before diving into that, I should emphasize that I'm definitely a pro sauna guy. If you go to Shortcut U at Substack, you'll see that I wrote an article called Sauna Science that I'll link below, where I break down a bunch of research on benefits of regular sauna use. And if you're really interested in this topic, I recommend checking out a review paper by Patrick and Johnson called Sauna Use as a Lifestyle Practice to Extend Health Span, because to date it is the most comprehensive and detailed review or paper I've read about the research around sauna, its benefits, the mechanisms behind its benefits, and different sauna protocols. Now, for this paper, the background leading up to this study is that there was some evidence suggesting that heat or increased temperature potentially interferes with spermatogenesis, or the process of developing new sperm in the testicles. And maybe the lowest piece of evidence is that the testicles typically are outside of the rest of the body on mammals, including humans. And one idea behind why this is the case is that we evolved so that the testicles could interact with the external environment, with the air, to cool down and maintain a temperature lower than the rest of our body. Quote, spermatogenesis is a complex, multi-step process involving the proliferation and differentiation of spermatogonia into mature sperm. In most mammals, testes need to be 2 to 8 degrees below core body temperature to work their best, and in most species, they reside outside the main body cavity. Many studies have shown that elevated intratesticular temperature has an adverse effect on spermatogenesis, and that extrinsic thermal stress to the scrotum may cause alteration of sperm parameters and apoptosis, end quote. And moving on in the introduction, the authors outline some of the evidence in animal models and cell culture models that show increased temperature interferes with spermatogenesis and related markers. Quote, previous findings in animal models have suggested that altered spermatogenesis, secondary exposure of the testes to high temperature, could be caused by insufficient blood supply and consequent hypoxic stress at parenchymal cells. In humans, scrotal heating of 1 to 1.5 degrees Celsius above normal has been shown to induce reduction in testes size, decreased sperm production, and altered sperm morphology, end quote. Carrying on a little bit further, quote, moreover, it has been documented that scrotal heating activates testicular stress responses common to other tissues, such as A, expression of hypoxia-inducible factor 1 alpha, or HIF1A, a transcription factor whose target genes are involved in vasodilation, angiogenesis, which is the process of growing new blood vessels, and glycolysis. B, HIF1A induced gene expression of vascular endothelial growth factor, or VEGF, a potent stimulator of angiogenesis and endothelial growth, acting through interaction with its respective receptors, the kinase insert domain, KDR, and the FMS-related tyrosine kinase 1, FLT1, and C, activation of heat shock proteins, which are in turn regulated by heat shock factors. Heat shock proteins are described as chaperonins with protective and anti-apoptotic roles, assisting the correct folding of nascent and stress-accumulated misfolded proteins and preventing their aggregation in response to thermal and oxidative stress. Increased expression of heat shock proteins and heat shock factors in sperm from oligozoospermic men, or men with decreased sperm counts, has been generally considered a testicular attempt to repair spermatogenic impairment and germ cell damage. A previous study by our group showed that heat shock proteins and heat shock factors are expressed in ejaculated sperm at different levels. In particular, we demonstrated that these proteins are upregulated in patients with oligozoospermia and varicocele, another issue with the testicles, I believe this one regarding blood flow, suggesting that this may represent a molecular marker of response to testicular damage, end quote. So that outlines some of the evidence leading up to this study that increased temperature leads to decreased testes size, decreased sperm production, altered sperm morphology, which is essentially abnormal shape of the sperm cells, as well as increased markers of pathways related to responses to cell stress. In particular, heat shock proteins, you've probably heard of when it comes to sauna use, are chaperone proteins, as that quote described, which are essentially proteins that facilitate the folding of strings of amino acids into proteins. So in the cell, when a protein is built, it starts out as a string of amino acids connected together. You can think of this like a string of beads, and then that string is folded in a particular way so that it is put into a particular structure so that different chemical groups on the different amino acids are showing in certain places or hidden in others so that it can interact with other molecules when it's carrying out its function. You can think of this kind of like Legos in that a given Lego has to have a certain shape in order to interact with other Legos. A given protein has to fold properly in order to have the correct chemical reactions when it interacts with other molecules. And chaperone proteins, like heat shock proteins, are involved in making sure that those strings of beads, those strings of amino acids, are folded correctly to begin with. And also, they're involved in refolding those proteins when they get unfolded or, as we call it, denatured. And this can happen if the temperature in a cell increases or due to some type of chemical damage. So that kind of sets the background for the idea that increased temperature would be negative for spermatogenesis. And the authors went on to test this, specifically to test whether or not sauna use in particular has the same effects in males age 30 about who haven't been using the sauna in the past year. Quote, 10 normozoospermic healthy volunteers mean age 33.2 were recruited in the study. The exclusion criteria were varicocele, metabolic syndrome, malignancies, history of cryptorchidism, sexual hormone alteration, and sauna exposure during the previous year. The Finnish sauna program consisted of full body exposure to 80 to 90 degrees Celsius, which, for those of you like me who are using the imperial system, I don't even know what it's called, but not the metric system, that translates to 176 to 194 degrees Fahrenheit, with humidity level ranging from 20 to 30% for 15 minutes, 2 days per week for 3 months. Scrotal temperature was recorded with infrared thermometer. The bulb was placed on the scrotal skin between the two testes and the testes pushed together until the temperature was stable. Temperature measurements were taken on the subjects before and immediately after each sauna session. Each measurement was repeated 3 times and the mean value was used. All the participants underwent blood withdrawal and semen analysis with 2 to 5 days of sexual abstinence at each point of the study. Before starting the sauna program, T0. After 3 months of sauna sessions, T1. After 3, T2. And 6, T3 from the months from the end of the sauna exposure, end quote. So they took these 10, 33-ish aged males and they had them use the sauna twice per week, following what I consider to be a pretty typical sauna protocol. And at baseline, after 3 months of sauna use, and then after 3 and 6 months post ceasing sauna use, they took some semen samples and blood samples, looked at a bunch of sperm metrics as well as some sex hormones, and they also looked at a bunch of markers of the different cellular pathways that I talked about when addressing kind of the introduction and the background leading up to this study. In particular, when it comes to the sperm metrics, they looked at sperm count, or how many sperm cells there were in a given sample, concentration, how many sperm cells per milliliter of semen fluid there were, the morphology, so the shape of the sperm cells, and viability, which, after reading a paper called Semen Analysis and Sperm Function Tests, How Much to Test by Bazan, I understand sperm viability to essentially be a test done when you have immotile sperm, so sperm that can't move, but that still may be functional in terms of being able to fertilize an egg. And this can be important or useful when it comes to something like in vitro fertilization, where we can take those sperm that are still functional but that just can't move and inject them directly into an egg to fertilize it. They also looked at motility, which, again, as I understand it, after reading a paper, I'll leave the links to these below, called Relationship Between Sperm Progressive Motility and DNA Integrity in Fertile and Infertile Men by El Bashir and colleagues. Motility can be broken down into three sort of categories. There's progressive motility, which I guess is considered kind of the normal category where the sperm can move linearly. There's non-progressive motility where the sperm can move, but they tend to move in small circles, and they also move slower than progressively motile sperm. And then there's non-motile or immotile sperm that cannot move. And one thing I found interesting when reading that paper was that there is some data suggesting that lack of motility can be related to DNA damage or oxidative stress within the cell, which kind of connects back to those stress response pathways that we talked about in the introduction and that the authors are looking at in this paper. Quote, progressive motility is the most important used measure of semen quality. The percentage of sperm that have forward progression is one of the most important prerequisites that affect both natural and assisted pregnancy. On the basis of the results of our study, decrease of spermatozoa progressive motility has been linked not only to male infertility, but also to an increase in sperm DNA fragmentation. This is in agreement with a study by Irvine et al. Has been reported a negative correlation of progressive motility and sperm DNA quality. During sperm maturation, human spermatozoa first migrate through the epididymis from testes with little or no motility. The percentage of motile sperm increases gradually as they pass through the epididymis. Thus, impaired sperm motility may occur due to high levels of oxidative stress caused by overproduced reactive oxygen species during the sperm maturation process, or a consequence of unbalanced apoptosis. Both oxidative stress and abortive apoptosis are also major causes of the generation of sperm chromatin damage in human spermatozoa. Thus, it is not surprisingly, in view of the above, an impairment of sperm progressive motility might be related with sperm DNA damage and, in turn, male infertility. End quote. So, like I was saying, that quote is interesting to me in that it highlights how potentially some of the markers that the authors in this study on sauna use and sperm metrics may be related to motility. And so if those markers are thrown off, we may see that progressive motility markers are also thrown off. So in addition to those sperm metrics, the authors looked at a bunch of different sex hormones as well as the cellular markers that we talked about in the introduction. Moving into their findings, quote, the mean scrotal temperature before and after the sauna sessions were 34.5 degrees Celsius and 37.5 degrees Celsius, respectively. Figure 2 reports the mean sperm parameters observed in 10 normozoospermic subjects before T0, after 3 months of sauna exposure T1, and after 3, T2, and 6 months T3 from the end of the sauna program. Figure 2a shows that both sperm concentration and count were significantly reduced at the end of the sauna program compared with baseline. 3 months after the end of the sauna program, T2, a reduced total sperm count was still present, while at T3 this parameter was completely restored. When considering other sperm parameters, we found a reduced percentage of the progressive motile sperm at the end of sauna exposure, whereas semen volume, sperm morphology, and viability did not change during the whole study period. End quote. So I'll throw figure 2 up on the screen, and what you'll see is that there's a pretty significant drop in sperm concentration, total sperm, and progressive motility after 3 months of using the sauna. The second thing you may notice is that, as the quote alluded to, there was no change in terms of normal morphology or sperm viability. And the third thing you might notice is that all of the metrics that were impacted returned to normal levels after either 3 or 6 months of ceasing sauna use. So it appears that, at least based on these metrics, that the impact sauna might have on spermatogenesis is temporary or at least reversible if you stop using the sauna. Moving on in their results, I'll throw up table 2, which shows the results of the male sex hormone, or just the sex hormone outcomes. And they looked at follicle stimulating hormone, luteinizing hormone, testosterone, estradiol, inhibin, and sex hormone binding globulin, all of which showed no statistically significant differences at any of the time points. Moving on to some of the cellular outcomes, or the outcomes related to the processes occurring uh, within the cell. They looked at, in figure 3, you'll see a bunch of different metrics related to DNA damage, as well as mitochondrial health and apoptosis, or sort of programmed cell death that occurs when there's significant damage within the cell. The cell sort of self-destructs, if you will. You'll see they looked at an AO assay, which is acridine orange, and that looks at sort of the status of the DNA within the cell. They don't see any differences there. The aniline assay, as I understand it, is a marker of sperm maturity and proper chromatin remodeling. So essentially, when the cell is, or the sperm cells are going through their processes of spermatogenesis, the chromatin, or the DNA, is sort of remodeled and restructured. And in the process, where the DNA is first wrapped around something called histone proteins, which is kind of like something that you can think of it like yarn being spooled around these histone proteins, the DNA being the yarn. Those histone proteins are replaced with other types of proteins, and essentially the aniline assay is a marker of whether or not that replacement has occurred. I'll throw a link to a study called Assessment of Chromatin Maturity in Human Spermatozoa, Useful Aniline Blue Assay for Routine Diagnosis of Male Fertility, which I read to kind of get some clarity on that assay in particular. And you see that after 3 months of sauna use, there is a modest, though statistically significant, decrease in the percentage, or the percentage of sperm showing normal markers as far as the aniline assay goes. And you also see a decrease in the percentage of sperm cells showing normal results for the decondensation assay, which, as I understand it, is an assay where they take the sperm cells and they induce a type of chemical stress that causes the DNA to break apart, and they see how many of these cells are able to withstand that kind of DNA stressor. If we look at the bottom half of figure 3, there are 3 more tests. There's an X and V assay, which, as I understand it, is a marker of apoptosis. We don't see any statistical differences there. The JC1 assay is a test looking at healthy mitochondrial function within the sperm. Again, I looked at a paper called Evaluation of Human Spermatozoa Mitochondrial Membrane Potential using the JC1 dye, and I gathered from that paper that the JC1 assay looks at the status of the membrane potential in the mitochondria in the sperm cells. So if you remember from like high school or college biology, neurons have a membrane potential where one side of the cell is, or has a higher or lower charge, electrical charge, than the other side. And the same is true of mitochondria. And the JC1 assay looks at whether or not that membrane potential is there. And if it's not, it's a sign of kind of abnormal or lacking mitochondrial function within those sperm cells. And you see after 3 months, there is a pretty significant increase in the percentage of sperm cells that are showing abnormal results as far as this marker of mitochondrial health. The Tunal assay, as I understand, is another marker related to DNA status, and we don't see any changes there. One takeaway I see from looking at these 3 markers that showed statistically significant differences is that all of them, similar to the sperm metrics, return back to normal after either 3 or 6 months of ceasing sauna use again, suggesting that the impacts of sauna use on these sperm metrics may be reversible. Quote, the AO test showed no significant variation of sperm DNA integrity after sauna-induced heat stress. On the contrary, histone protamine replacement and chromatin condensation, tested by aniline and decondensation tests, displayed a significant impairment at T1 compared with T0, 69 versus 78 and 63 versus 70. Those chromatin alterations completely disappeared at 3 months from the end of the sauna exposure. At the same time, the evaluation of molecular markers of early and late sperm apoptosis documented a significant increase in the percentage of sperm with impaired mitochondrial function compared with basal, both at the end of the sauna exposure and up to 3 months from sauna suspension. In contrast, markers of later stage apoptosis, the NX and V assay and the Tunal test, showed no significant variation during the whole period, despite an apparent increase at T1, end quote. Moving on to the final results of the study, we have figure 4 that looked at HIF1 alpha, KDR, FLT1, and VEGF, which are the markers that we were talking about in the introduction of responses within the cell to stress. And in particular, I should kind of explain that we're not measuring these molecules or these proteins in particular. What we're measuring is the DNA expression, so levels of RNA and DNA, suggesting how the activity of these proteins, or how many of these proteins are being built within the sperm cells. And that is kind of a proxy for whether or not these sperm cells are responding to stress within the cell. And again, what we see, at least for HIF1 alpha, KDR, and FLT1, is that there's a huge spike, or a pretty significant spike, following 3 months of sauna use. And then after 3 or 6 months of ceasing sauna use, those markers return to baseline, or interestingly, below baseline when it comes to HIF1 alpha or FLT1. The other interesting finding from this figure is that VEGF, which is a marker or molecule related to increasing blood vessels in the area, doesn't increase after 3 months of sauna use, but then 3 months after ceasing, we do see an increase in terms of the kind of proxy for VEGF production within the cell. Moving on to figure 5, we are looking at heat shock proteins and heat shock factors, which again are essentially molecules associated with responding to that cell stress, particularly with denatured proteins, which can come about to increase temperature, and refolding those proteins correctly. We see, similar to the other patterns, an increase after 3 months of sauna use, and then a return to baseline levels after 3 or 6 months of ceasing sauna use, or at least a significant drop from the significant increase following 3 months. Quote, gene expression of hypoxia-related factors evaluated in ejaculated spermatozoa at different points within the study is reported in figure 4. HIF1A, KDR, and FLT1 showed a spiked increase after the end of the sauna program, T1. Thereafter, at T2, they had a rapid reduction, and at T3, HIF1A and FLT1 showed lower levels than T0. Interestingly, VEGF expression displayed a similar but delayed trend with a significant increase at T2 and a prompt reduction at the end of the study period. Gene expression analysis of heat shock proteins and factors observed in ejaculated sperm along the study is shown in figure 5. All these genes displayed a significant upregulation after the sauna exposure, T1. Thereafter, heat shock protein 90, heat shock factor 1, and heat shock factor Y had a swift return to baseline levels after the first trimester of recovery, while heat shock factor 2 showed a higher expression up to T2, and heat shock protein 70 up to T3. Unlike HIF1A and FLT1, the trend of gene expression of heat shock proteins and heat shock factors during recovery times never showed lower levels than basal. End quote. So that wraps up all of the results for the study. Before getting to kind of my takeaways, I'll run down the author's conclusion. Quote, in conclusion, our data demonstrated for the first time that in normozoospermic subjects, scrotal hyperthermia induced a significant but reversible impairment of spermatogenesis, including alteration of sperm parameters, mitochondrial function, and sperm DNA packaging. Moreover, genital exposure to Finnish sauna resulted in a complex gene response, including expression of genes associated with heat stress and hypoxia. The strong upregulation of these genes observed in men after sauna exposure suggested that it might represent both a molecular marker of stress response and an attempt to preserve spermatogenesis from testicular heating. It is not clear whether our findings are related to elevation of testicular temperature or to side effects induced by heating of whole body. Whatever the large use of Finnish sauna in Nordic countries and its growing use in other parts of the world make it important to consider the impact of this lifestyle choice on men's fertility. More studies are needed to understand whether the same protective mechanisms observed in normozoospermic men are fully operational in subfertile men and in prepubertal subjects in whom spermatogenesis is at a more vulnerable developmental period, and whether they can protect such men from testicular damage. End quote. So a couple of things to highlight as far as that conclusion, as well as a couple other caveats when it comes to this study. First, as the authors alluded to, we don't know per se if the results of the study are due specifically to increases in the temperature of the testicles via the temperature increases we saw when they saw when measuring the scrotal temperature, or if it's just a general increase that comes from heating the entirety of the body. In other words, if they prevented the increased temperature of the testicles but heated the rest of the body, would we still see these same results? We're not quite sure based on this study alone. Another interesting point that they make is that it's unclear whether or not the results we see here apply to individuals who have infertility or who have issues with fertility, as well as boys who haven't hit puberty yet. It's unclear whether or not, as we see in this study, it seems that the impacts of sauna use or elevated temperature on sperm metrics is reversible after ceasing sauna use, is applicable to individuals who are more susceptible to infertility or individuals who haven't hit puberty yet. In addition to that, I think some interesting or important caveats to mention is that this is only a single study done in just 10 subjects, and the subjects hadn't used sauna for a year and they only used sauna for 3 months. So it's unclear whether or not this applies to individuals who have been using sauna for years, or whether or not individuals who haven't used sauna use sauna and then continue using it for longer than 3 months. It's also unclear the exact timeline of the results we've seen, in that they only measured these metrics every 3 months. So it's possible that there was a nonlinear pattern, or that the metrics returned back to normal, for example, before 3 months or 6 months, if they looked at 1 month after ceasing sauna use, 2 months, 3 months, 4 months, and so forth. We may see a different timeline or a different exact points where the metrics return back to baseline or not. With those caveats kind of taken into account, I have two sort of big takeaways looking at this study. Before I talk about them, I should emphasize I'm definitely not a doctor, definitely not giving you medical advice, definitely not telling you what to do or what not to do if you're looking to have a baby or trying to avoid having a baby. These are just kind of my thoughts on the data and the study. When it comes to individuals trying to have a baby, sauna use may be an important factor to consider, in that if the male in the relationship is using the sauna, it may be impacting their sperm metrics and potentially their fertility or the probability that they'd be able to get their partner pregnant. Again, this is just going off of one study and this is just kind of my extrapolation, but I have heard, some well-respected experts in the field, like I think Dr. Andy Galpin, I think Dr. Andrew Huberman, Dr. Kyle Gillette, I think Dr. Rhonda Patrick, who's the author of that great review that I talked about at the beginning of this video. I think they all have mentioned that it may be a good idea to stop using the sauna if you're trying to have a baby. Don't take me on my word for that, but I'm pretty sure that they've all referenced that. Just them saying it doesn't make it true, but just to kind of back myself up here with this point that I'm making. My second takeaway is that for individuals who are trying to avoid having a baby or getting their partner pregnant, again, I'm not a doctor. I'm definitely not telling you what you should or shouldn't do, or telling you that sauna use is a perfect contraceptive. But one thought I have is that if you're trying to avoid getting pregnant or getting your partner pregnant, sauna use may kind of be an add-on insurance plan to additionally decrease the probability of that pregnancy occurring. Not saying it's a contraceptive method all on its own, but maybe it would add a bit of peace of mind on top of other contraceptive methods, just to, again, decrease that probability of pregnancy occurring, based on this study where there's a significant decrease in sperm count, sperm concentration, and progressive sperm motility, all presumably important or meaningful metrics when it comes to male fertility, as well as some of those other metrics that they looked at in terms of pathways within the cell showing responses to cell stress, DNA issues, and what have you. And that pretty much wraps up my thoughts on this study. If you want more content like this, or you want to support Shortcut U, you can go ahead and subscribe on YouTube or at Substack. And if you prefer kind of more concise, organized content, you can check out kind of the deep dives and other articles I write on Substack, because I think they're a lot more concise and organized than me just babbling off and going off on tangents in these videos. So if you're into that type of thing, those might be something worth checking out. Otherwise, I will catch you next time.